Thank you, Ashley. Well, I don't know. I really thank you for coming, and I don't think this crowd, it is great. You have 36 people showed up. I really appreciate. Tea, I don't know. Everybody just drink tea for lunch? The sweet tea there, right? So it is new, and brand new for us. And in terms of industry, it's even infant. So we need to go there. I'm Dongyin Zhang, and if you have any questions or want to talk about the tea, you can write an email to me or make a phone call. This is my information here. It was also, I put this handout on the web page so you guys can download it from those conference web page. Right? And tea is majorly excellent for the garden plants. As you say, you have a big leaf, small leaf, triggered leaf, yellow, and purple, right? All kinds of stuff. It's really nice in the ornamental plants. But today, I really want to talk about tea as a commercial crops, right? Thank you very much, Dr. Tim Coulomb, for this opportunity and the conference. Actually, give me the opportunity to share with you. Relatively speaking, tea is new to us, really. And we get a lot of tea because from foreign countries, not from here in the United States. Tea is really growing well here in Georgia. As you notice, tea is probably popular drink in the world except the water, right? Actually, when you talk about back to our country, right, without the Boston Tea Party or events, we don't have country. So this is really important. Doesn't matter commercially or historically, it is right. It is very important. So far, only production tea company is Charleston Tea in the continental USA. Now, recent years, there have many small growers are beginning to produce tea. The big challenge so far is the import tea is too cheap. We want to produce something can compete with it. This is probably one part. Actually, we did a lot of tea research, including USDA, former tea research center. They collect hundreds of cultivars from around the world. It's amazing. This is established in 1942. Now it's abandoned. Tea is really demanded by U.S., right? As you can see here, the marketing and the export is a lot of money there. I talked with some tea growers, small growers in Mississippi. They said if you can sell for $15 per pound, you can make some profit, $2 or $3 per pound. But still, remember, this is special. The import so far, anybody knows how much import tea price? $4 per pound. So this kind of compete, but it's different. The tea is not as good as what we like. This is probably. Anybody drink the tea in Trump Hotel? I tried once. $55, right? What do you get? A cup of tea and dessert. The cake. It is really, they can market very high price if we know how to market it. So don't ask me about marketing questions. Together, we build the industry first. Then we can go from there, right? Of course, southern part, when I first come back and interview for the job in Georgia, my host said, what do you want to eat tonight? I said, of course, barbecue, right? You have iced tea, you have barbecue. This is our staple food, so popular. So what I want to share with you a little bit is, number one is propagation of tea is not too difficult, right? I propagate thousands of teas, right? 1 year I propagate 10,000 of tea. It's just amazing. Amazing. It's easy to propagate it, especially in October, September, October, November. Those 3 months, take tea cuttings, you can grow in them very well. You don't use hormone. If you want to use hormone, would be hope better to grow in those. This is the tea, we produce it. Right before pandemic, this is how many tea I produced. It's just amazing, right? I think about is everything's good. Turning out, pandemic coming, you don't have any hope. I don't know what to do with all the teas. What do we do? I put it, majority of them is not wasted, but I still plant some in the Athens, Georgia, right? The rotten well, about to take 2 months, 2 months to root it, and then another month or two can be get better. So tea, I did a lot of work on the tea growing. If you just sell tea as ornamental plants, you still can make very good profit because you can take the cuttings in September, October, then grow into next year. Next June or July, you can sell the tea for $19, which is pretty good price. So this is what I say is, different tea have a different property to root it. So I don't want to go details, but those is the property you get. Different clones have different root ability from 25% all the way to what? To 100%. So this is a property. And to summarize a little bit of routine, I can tell you very confidently that tea is a lot too difficult to propagate cloning. So we will try to do some work and select a better clone for Georgia. This is our next step. It's very, very important. Also, if you want to grow tea a little bit faster, you can take longer tea branch cuttings, right? For 5 loads, you can grow in a little bit more. Relatively, it's good. It's good. So this is probably, and number of loads per cuttings will make later growth very, very different. So this is probably, this is tea propagation facility, and I was supported by USDA, visit China production, tea production, and they're really big. They're really big. This is a garden in the horticulture farm right nearby the road, and I planted 2 years ago, not the last year before. This is how they look in the tea garden. Relatively, I want to deliver it easy to propagate, and also Georgia weather is very good for growing those tea, right? 2 types. One, tea loves this red clay for some reason, right? As long as you can get irrigation, they're very good, right? This is a property. Second part is if you're sandy soil, you have to make sure irrigation is follow. They still don't demand. They don't demand. So this is a property. I really think Georgia is a great place to produce, to growing tea. This is some conditions I want to share with you. But from those all the way to North Georgia to southern part of the Georgia, you can growing tea well, right? Some of you can always remember your organic sections, right? We want growing organic tea. When you prepare the field, it's worth it. You put all this organic fertilizer on the trench, right? This is my suggestion. So far, if you look at those on this side, you can say you can plant 2 rows, but zigzag stock planting about 1.5, 1.5 inch, or what the feet. So about 18 inch apart from the plants. So the row should be less than 18 inch because of the height of the equal length triangle. So this is probably organic fertilizer, apply for it, and you're allowed to regulate it. If you plant with a bunch of organic fertilizer, you don't need to take care of fertilizer for a few years. Tea will be growing very well, very well. So this is the one part is, we did growing a tea evaluation garden right in the horticulture farm. This is planted last year. This tea garden is selected 10 cultivar we selected. We want to try those cultivar, whether they're doing well here in Georgia. So for example, if I ask, right, Dr. Coulon or me, what kind of cultivar I should grow in? We cannot give an answer. But a few years later, we definitely give you an answer and make best tea available to Georgia growers. This is our research will be leading to. Also, if you want to apply fertilizer later, what you do is you get a cow manure or this organic stuff, trench on the side, and put it in. The root should be able to take it, right? So, generally speaking, Georgia we can propagate tea, can grow in tea well and organically. There's a lot of marketing strategy, right? You can put those organic on it. Then you think about a little bit, I will go through a little bit, how to harvest the tea. Agriculture business, okay, you cannot use it manually. What do? you have to go to machine, right? Just our students doing this. Is half the tea by hands, but actually you have to use a machine. If it, with machine, without a machine, you cannot make it, right? A lot of concept I want to give you idea is, people said, can I grow in red tea, black tea, or some green tea? So don't mix it. Any tea you can make different tea. One tea cultivars, but some tea is better, some tea is worse. But anyway, try to use the one tea, the procedure different will make a different tea. So I just go quick about this part is to make sure you do have those different type of tea. I make it. The last part I want to share with you is our research, right? The better research for better tea. This is a professor in the genetics at UJ. He wrote for Nature. He talked to me about those, and we talk about those. It's very important. Tea is kind of plant worthy, can harvest all year round. Don't think like a blueberry at one season, peach one season. No, here is it can harvest from spring all the way to the end. You probably 13 to 20 times a year. So the harvest. If you lose the one for the little frost, it's fine. You can get it. What we try to do is we will try to select better cultivars and also do the triploid breeding. So tea seed mature from October all the way to next October. Useless, right? Take a lot of energy. What we try to do is we try to breed in triploid tea. Hopefully we can get some triploid tea for you guys to grow in without worry about nutrients level, right? Those research part I don't have time to share with you, but this is what we want to do. I really think about tea is a great potential crop for Georgia commercial growers. And we need to work together to make this happen. It's easy to grow, not too much investment, and we need to get together and come here to do those and make sure we will have someday, we have Georgia tea, right? We have Georgia growing, we have Georgia tea so far. I don't have time to get all this other stuff, but if you have any questions, just talk to us, actually. And also, this talk is available on this conference website. You guys can have access for it, and then the tapes. I really want to thank you, our lab crows people, and the University of Georgia, and especially the Research Foundation support this research. Hopefully someday we all drink what? Tea from Georgia. Okay, actually, you don't give me too much time. yeah. Thank you very much. You have a 46 people. It's good. Yes.
So your slide there, you talked about harvesting. Is it one leaf and a bud when you go through? Was that correct? I just missed it.
Well, when you harvest the tea, you can just harvest tea bud. This is higher quality of tea. You know how many tea bud make 1 pound of dry tea? 80,000. This means you need to pick up 80,000. How much time you want to put on it? So majority of people will take 1 bud and 1 leaf. And then 1 bud, 2 leaves. All of those are pretty good quality tea. So it depends on the quality, and the people will make it different. The price goes way higher for those. Yes.
How many times are you trimming your plants per year?
Well, this is what I say. When you started the plantation, we started pruning it at 15 inch. When the tea gets 15 inch from the ground, pinch it. 20 inch, pinch it, all the way to 40 inch. Okay. Then for the 40 inch, you don't need to trim it. You just harvest. You harvest about 13 to 40 times, to 20 times a year. So it depends on how it grows. Yeah, you don't need it anymore. Yeah.
What's the ecosystem that where tea is native?
Oh, he said China.
Now, you mentioned.
It's very similar to Georgia climate. The area, the average temperature. The only difference, our Georgia temperature always have late frost.
I was thinking shade trees,
Well, you need some shade is better.
Pines to oaks to pines.
Well, don't get those big trees. You want to get some tree can shade a little bit to the tea will be great. But if you don't shade it, you need enough irrigation to keep the tea growth. So partial shade will be great, but not required. You have to get irrigation system. Even in the full sun, they're still doing good in the summer. We have those in the full sun. They're pretty
Would you
good.
put a percentage figure on it, like
Well,
60% shade?
70, 60,
40%?
you are 30%. 30 to 40% shade will be perfect. Yeah, this is what our result. This is from other school. They did shade study. Yeah. So the only thing advice for you guys is Georgia winter. I have learned those late frost kill plants, not just damage plants, the killed plants. So, but in tea, I think it's pretty good. You can kill it once. If you can harvest before the frost, you can. But if you don't, just lose one crop. Next time they'll come back again. So it's pretty nice.
And if you have any more questions, oh, we have time for one more question. Yep.
What would you say is a major pest for us?
So far, I don't have anything there, but I'm sure that definitely after you grow it, you have something come up. So far, I never have anything. They're pretty good. I just go there, pick up the tea, and eat it myself all the time.
Tea. I don't know what.
Well, thank you so much, and we will make sure you have access to his information. If you're interested in that, definitely contact your local extension, and they can get you in touch with Dr. Zhang. So thank you. Our next speaker is Dr. Jake Maurer. He's the Extension Associate Professor at Texas A&M University. And Jake provides leadership and education in the areas of soil fertility and water resource stewardship. So today he's going to talk about sustainable management practices and soil nutrients and refining our understanding of tillage, residue, and fertility management to achieve goals in sustainability and profitability in food and fiber producers. So, is yours on? Yours is on here.
You found my website, huh?
Covered crops and pea, right?
Yeah, that's the one.
So this is the first of Dr. Maurer's talks. He will be our final speaker. So,
All right. Second and final.
yeah.
Okay,
Yes.
thank you, Ashley. I appreciate it. Okay. Am I being heard? Yes? Okay, good. All right. My name's Jake Maurer. Last name rhymes with flower. I know there's a sneaky R in there. Don't worry about it. Just remember flower. If you want to give me a call, talk to me later on today. That's how you pronounce it. Been that way my whole life. Right now I'm going to talk about cover crops and increasing phosphorus availability in soil for basically any crop. Okay. When I got to Texas A&M in 2015, I went out and toured our research farm, and there was one person using cover crops out there. I had moved from the University of Georgia out there, and there was a lot of people working on cover crops here. So I was kind of confused, and that was where I decided, all right, here's a gap in what we're working on. I'm going to attack it right now. And I think a lot of our farmers had a lot of questions about it because they're getting a lot of information on the web. And we didn't have anything really good in extension to tell them that was very detailed. So I just started planting all the cover crops everywhere I went, working with farmers, and I identify as a soil chemist. So I just started measuring everything about the soil and seeing what popped up to me, what benefits could we get. And so as a member of the Southern Cover Crop Council, I will tell you that we always emphasize knowing what you want to get out of a cover crop before you go getting into cover cropping. What do you want? Nitrogen, biomass? Do you want to build soil function? Do you want to put carbon there? Do you want a pollinator habitat, et cetera? But and I remember talking with someone from the Northeastern Cover Crop Society or Council, And they were really not a believer that nutrients other than nitrogen could be provided or could be increased or enhanced by cover cropping. And we showed him, one of our graduate students, research, which I'll show you here in a little while, and he was made a believer. He was like, I always thought you couldn't do that. And so over the last few years, I've really found that phosphorus is something we can target increasing from a cover crop. So does anyone here believe me? Do you not believe me? Should I can go forward? Okay. Y'all are really responsive. I get some people in classes after lunch on campus sometimes this responsive. Anyway, this is how our talk is going to go today. We're going to talk a little bit about the soil phosphorus cycle. And just a refresher, I don't want to insult anybody, but we're going to make sure we understand a little bit about it. Cover crop potential to increase soil phosphorus will be our next thing, and then I'll just talk with some research that I've done personally out at Texas A&M, and then we'll summarize everything for you. There you go, you got to hit it twice. So this is a really simple diagram of the phosphorus cycle in soil. I'll give you 10 more seconds to memorize it, and we'll do the pop quiz. Okay, so really what I'm most concerned about, I'll just stand near it and we'll get. Really what I'm most concerned about, I think, for growing vegetables, fruits, any kind of crop, is this right here. This is the plant available phosphorus. It is a tiny amount in the soil solution at any given time. It can be really frustrating under many conditions to deliver phosphorus to your crop, whatever it is. But it's going to exist in inorganic forms and organic forms in the soil that will continue to feed back into that solution as plants take it up. That's what we hope for. And our inputs are going to be things like chemical fertilizer, cover crop residue, and manure, biosolids, organic fertilizers, right? And I think for organic growers, this is really, really important. Like, how can you harness the energy of a cover crop to increase the phosphorus that gets to your crop, right? Because sometimes if we're using manures, we may be over-applying phosphorus, right? Is anybody aware of that? It's not balanced? Okay, so it can be, you know, there's a lot of poultry litter in Georgia. I did my entire dissertation on poultry litter. I grew up on a chicken farm. It's a real thing. But you can over-apply phosphorus very easily. You can have environmental harms. You can cause something called phosphorus-induced zinc deficiency, where your plant can't get zinc up the stem to the leaves because the phosphorus is complexing it in the stem. So there's a lot of frustrations with phosphorus in many different systems. We've got the inorganic side and the organic side. I think if you've got a sandy soil, you're probably not dealing with too many complexations on the inorganic side. But if you have a red dirt or a better alluvial soil near a river, probably some of this is going on. So it can get locked up a little bit away from your plant, your crop, even if you apply chemical fertilizer. But as it's released from manures, it's got to be broken down into phosphate, PO4, for the plant to take it up, right? So, you know, there is no pop quiz. The point is, it's a really complex cycle. And I think the hammer drill is helping me out here. Just making this more impactful. Anyway, if we focus on the inorganic side, there's a lot of clay complexation. If you have a sandy soil, this isn't a very big deal. But when we're trying to liberate phosphorus from these complexations on the inorganic side, with things like plant roots are releasing phosphatase enzymes that will break down phosphorus. There's a modification of pH that can adjust soil pH around the rhizosphere to something that's more favorable for phosphorus solubility. And there's uptake and temporary removal from soil reaction, like by putting it into plants, right? It's in that tissue, so it's sort of saved from getting complexed and taking away from that plant or future plants, right, until it's broken down again. And on the organic side, we've got temporary incorporation into microbial biomass. We've got, we can take advantage of arbuscular mycorrhizal fungi. Anybody heard of these symbiotic organisms? They're really fascinating. I study them in the lab all the time and in the field. They are famous for acquiring more phosphorus, right? And they need a living root to host them, right? And so if you don't have a cover crop in your off season, you will not be supplying a habitat for arbuscular mycorrhizal fungi. And then finally, we can just flood the rhizosphere with sugars, right, that will attract chemotaxic bacteria closer. They'll release their own phosphatase enzymes. They'll do other things to liberate different nutrients, including phosphorus in the rhizosphere that benefit the plant with a small outlay of photosynthetic product in the form of carbohydrates and sugars to feed them. It's like it's bait, you know. They're fishing for bacteria, these roots. There's a lot of fascinating stuff going on. And phosphorus is also famous for not being soluble except for between like 6.2 and 6.7. As you move away from those pHs on either side, you really limit the ability of phosphorus to come into soil solution. So again, it's just a lot of tricky stuff to work with, but harnessing plant power and some of the micro critters that are in the soil can help us access that phosphorus a little bit better. So there's a lot of interest in biotechnology that's increasing rapidly among farmers. I mean, there's a lot of biotechnology products here. There are inoculants, biostimulants. A lot of that has a good basis in the science. But arbuscular mycorrhizal fungi, they naturally form associations with like 88, 90% of the plants out there, just not brassicas and apparently not buckwheat. So when I dig up clods with my students out in the field, I think this is the wheat field, that we will look for evidence of these fungi, right? Am I out of time? I'm just kidding. Yeah. Are you going to find the hammer driller? So those are mycelia or the hyphae that are coming out, and hopefully, you know, when I stained these roots, I did find evidence of hyphae inside the root and storage structures called vesicles and transfer structures called arbuscules existing in these wheat roots. So a lot of fun to find that. We find that they are associated with less tilled soils, greater organic matter. If there's more organic phosphorus in there, we'll see a lot more of these arbuscular mycorrhizal fungi occurring. And so we find a lot of relationships with that. There was this famous, or at least in my lab, a famous research project back in 1998 that showed that if you had wheat over the winter, you could increase the beneficial infection in corn, increasing the kernel weight or the yield of grain in corn. It was closely associated. And what I really like about this study was they separated these 4 treatments: no cover and tilled, which is none of the recommended practices for building soil function, right? No cover crop and tilling. Y'all get that right? Okay. I know it was good fried chicken, I know. Cover cropping and tilling, which is one of the 2 recommended practices, and then no cover and no till, and then cover cropping and no till, right? So that's 2 practices combined that we recommend, none of them, and then the intermediate one only, right? And you can see what's really fascinating to me is that with no cover, no till, all of the yield and infection grouping is all way down here, right? As we go to no cover, no till, we move up a little bit, right? Can y'all see that? All right. As we have cover but tilled, they're grouped up here. And if we have both things happening, they're all the way over here with more yield, more infections, right? Beneficial infections. So what this tells me is that having a living root is more important than refraining from tilling if you want to have arbuscular mycorrhizal fungi benefits, right? Now, it is a symbiosis, it's a mutualism, so if there's no need for mutualism, you don't have a lot of infections, right? But you can stimulate this by reducing the amount of phosphorus fertilizer that's added, and you will see them pick up the slack and take up more from the soil. So we did one winter, I got a poorly funded study from the Wheat Board in Texas, but it was enough to sort of look at this idea. Like if I inoculate with arbuscular mycorrhizal fungi, and I have tillage and I have no tillage, But this time, wheat is the cash crop and buckwheat is the summer cover before it, instead of the previous research that I had shown you. So we did this. Unfortunately, our planter didn't get the wheat seed in on the no-till, so we only have tillage and cover and no cover to look at here. So this is what buckwheat looks like. Have anybody seen this before? Who loves a good buckwheat pancake? Okay, it's a really interesting crop. Yeah, it's a great flower. Yeah. So, anyway, it's an interesting plant. It doesn't grow very high. It has a really nice, modest, but pretty white flower. But it really does amazing things to the soil. So I recommend it for the southeast, from Texas all the way to here, North Carolina, for a summer cover crop. So if you're growing kales, broccoli, something that's a winter cool season, you're planting onions, you might have this before that kind of crop. In the summer, it will grow pretty much all the way till the first frost, right? And a fun plant to grow. So we measured grain yield, protein, biomass, a whole bunch of things, but really what I'm going to cover is the phosphorus. But I have to say that where we had buckwheat, we had a significantly measurable ding in wheat grain yield, which is not what you want to be communicating to farmers, right? So I have to be upfront about that. How do we figure it? I did not fertilize with phosphorus at all, though, right? So that was a little bit vexing, I guess is the polite word for it. We found no significant difference between cover crop and yield. We did find if we added fungicide to protect the wheat first, which is not going to be an organic thing, we did have better yields. Grain protein was significantly different, and with the cover crop and with A&MF inoculant. But what just floored me was the phosphorus outcome, okay? Wherever we had buckwheat, we almost doubled where we didn't have a previous buckwheat cover crop in whole plant phosphorus. I've never seen anything that stark, that obvious. We didn't fertilize with phosphorus. The field needed it. This buckwheat did something to the soil that we made a huge difference. So we had taken some root samples of the buckwheat and the wheat, and they're inoculated with arbuscular mycorrhizal fungi, and we looked to see for evidence of arbuscular mycorrhizal fungi, and we didn't find any. Buckwheat doesn't really host it very well, and we didn't find any in the wheat either. So I talked to our wheat breeders about why they had bred the wheat away from associations with AMF, but they didn't laugh. Anyway, that's pretty stark, right? Every single associated treatment where the only difference is cover cropping and no cover cropping with buckwheat, we had twice as much whole plant phosphorus. That was pretty amazing to me. We also had numerically greater whole plant nitrogen, but not significant. So this tells me that we really need to follow up on this research and find out what's going on. But if we didn't have arbuscular mycorrhizal fungi, can anybody tell me what might be another mechanism for increasing the phosphorus? I gave you some hints earlier. It's okay, you can raise your hand.
Microbial solubilization of phosphorus.
Cool, okay. Yeah, that's a great response. Maybe. All right, so I had a grad student working on this. She came, they're supposed to work when classes aren't in. Classes start on Tuesday. So she came back and got some really good data for me and sent it to me while I was in the airport a couple of days ago. And actually where we had cover crop, we had a significantly higher amount of phosphatase activity, an enzyme released by bacteria and roots to liberate phosphorus. Now, I'm going to be also clear and upfront with you. This isn't a big difference. It is significantly different. Like we could separate them statistically, but the difference between 1.08 and 1.2, I don't know that it's all that practical because the ranges you'll find out there are anywhere from 0.1 to 20. Okay, some people have said 80 micromoles per gram of soil per hour, but that tells me this is something I want to look into a lot further, right? So if buckwheat won't host AMF, are those roots really releasing a lot of something that solubilizes phosphorus in the soil solution and liberates it for the next plant? So how are we doing on time? I have 10 minutes? Okay. So that was really fun. We're going to follow up on this. I'm going to grow buckwheat in the greenhouse. I'm going to grow it in the field, and we're going to look at more of these oats, wheat, and onions coming up in the future. So that was really fun. But what if you're growing a summer crop? Is there a cover crop for the cool season that will help you get phosphorus out of the soil? Yes. Yes, there is. I'm glad you asked. All right. So we were working in organic cotton, and we had cereal rye as a cover crop. And these are some friends of mine who are weed scientists. They were really interested in the weed suppression of cereal rye, which it's famous for. It's also known for phosphorus acquisition. But I was on the committee for the student, and I suggested that he do some more intensive soil sampling and analysis, and I really wanted to look at what the phosphorus was doing. So this study was about seeding rate of cereal rye and date of termination. So basically, we would be planting cotton in Texas around mid-March, okay? Maybe first week of April at the latest. If you have a really rainy spring, June, but then you're harvesting in December. So we planted October cereal rye, and it grew all winter, and then terminated like mid-February, end of February, and the middle of March. So 2 weeks apart each time. And then the seeding rates are 0, 18 pounds, 35 pounds, and 70 pounds per acre, right? And the thing that I want to mention is that a lot of times the older or early advice on seeding rates for cover crops was quite heavy. 70 pounds is more than a 50-pound bag, right? But we were finding that you could get away with things closer to half a bag and get the same type of biomass and the same type of benefits. So looks like if you can let 18 pounds go until the last minute, you can get some pretty good phosphorus liberated from the soil, and it shows up in the soil test, at least the one we use in Texas. You use a different one in most South Carolina and Georgia. But if you go to 35 pounds, you get even more of a benefit at that earlier termination date, which gives you more time to, do everything you need in the field before you plant, right? So, and then going out to 70 got you more at that earlier, but not at the later, right? And so I'm thinking, depending on how long you can let it go, it's either 18 or 35 pounds. But that, according to our soil test, was leaving between 70 and 100 pounds of P2O5, the fertilizer equivalent of phosphorus, per acre, more than no cereal rye was. And that's showing up in the soil test, all right? And with the buckwheat, we weren't seeing it in the soil test, but we were certainly seeing it in the plant uptake. And this, with the cotton, I don't know that we saw it in the plant uptake because they didn't have enough money to do whole plant nutrition, but we are seeing real benefits to soil phosphorus liberation for your next crop using certain strategically selected cover crops, right? So our takeaways here are that using natural plant functions, even if it's not phosphorus you're after, using natural plant functions from cover crops is a great way to manage your farms, right? Whatever you're growing. But we know that we can improve soil phosphorus availability. It's definitely possible. Targeting this activity might cause reductions in other desirable goals. Everything is not a win-win in this world, right? But that research needs to be developed. So we need more of this research to harness it in the right direction without losing the thing that makes us money by spending a little less on phosphorus, right? And so then my final recommendations for you all are going to be, if you're growing a winter cash crop, put in some buckwheat at about 20 pounds per acre in the summer. All right, it's a pretty plant. You can feed it to animals. You can leave it there. You can till it in, green manure it. Over the cool season, cereal rye will also help out with pea. Now, the one caveat to that is when we repeated the organic cotton study with cereal rye before and the next field over, It was a little clayier, and we didn't see statistically separable results. And so I think that cereal rye is going to be more important for sandy soils than for soils with a little bit of clay. Fortunately, the Atlantic coastal plain is blessed with sandy soils, right? And then for buckwheat, that was actually in a silty clay loam soil, so there was plenty of clay there. We haven't repeated this in different soil types, but that is one of the next things for me to do this year. So that is my last slide, and I'm pretty good at that, yeah, okay.
Do we have
So-
any questions from the audience?
Yeah.
Okay, perfect. I'm going to go ahead and introduce our next speaker. We have Awardatu Bukhari. She is a postdoctoral associate from the University of Florida in the Department of Plant Pathology. Awardatu is working on the efficacy of essential oils as control for fungal and bacterial plant pathogens of peach and blueberries. So I will turn it over to you, and thank you very much for being here.
Afternoon, everybody. Sorry. I lost my voice yesterday, so I hope you can all hear me. All right, let's get started. So I'd like to start with thanking our support institution. This work was not possible possible with the help of the USDA NIFA grant. And the work is part of a multi-state and multi-crop grant. So we look at mango, avocado, blueberry, and peach. And we have collaborators in Florida, California, Hawaii, Georgia, and South Carolina. And I'd like to acknowledge all the people that made this work possible, especially our industry partners, mainly for blueberry, Island Grove, ag products, and for peach, McLean family farms. Without them, we will not be able to actually put the essential oils in organic farms to see if they were efficacious or not. So we really needed their help, and they were there to help us out with that. So, all right, so let's get started. So, as many of you know, possibly all of you, fruit crop production is constantly challenged by plant diseases. And these are just a few of the blueberry diseases that we were concerned about in Florida, and we wanted to look at. And here are some of the peach diseases that we wanted to look at. Mostly for both blueberry and peach, rust is a big deal for us in Florida. For peach, fungal gummosis, as well as brown rot, which is caused by Monilinia fruticola, which is a fungal pathogen. So for all the diseases that cause problems or are challenging fruit crop production for blueberry and peach, we also do have products or materials that we can use or growers can use in the field. So, and you may be familiar with a lot of these, and the materials are divided into many different groups. Some of them are part of the natural group, mostly made of essential oils. And plant essential oils are broad spectrum activity material. So what that means is they don't only help, they are kind of antimicrobial, so fungi and also bacterial antifungal and antimicrobial, meaning bacterial. And also, we have seen some effects on soft body arthropods. So they also have no negative environmental impact, and they're relatively safe for human consumption. So for all these characteristics, they've been used as an alternative to synthetic materials in organic, and but also you can use them in conventional farming. So, but what are these? Well, these are complex mixtures, substances from plants. So the process of these, you start with your raw materials, which are plants. For us, we use savory, oregano, and thyme. And then you go through the process as shown on this picture. And in the end, these are the type of oil or product that you have. And for farmers, you're mostly familiar with these type of products, which are commercial formulation of the products I just showed you. So you see, you take the pure oils, which then we add other ingredients to make them soluble in water, and then that makes them biopesticide formulation that you can then use on your plants. And for our purposes, we use and interested incinerate, time guard, and Timeworks Act. So let's go through our materials and methods really quick. So our main question was, what's the efficacy of these essential oils to actually control the fungal diseases of blueberry and peach? So we had 3 main phases to answer this question. So the first one being the lab assays. So what we did was poison assays. Pretty much we had 5 different, 5 products. We had 3 pure oils, thyme oil, as I said, savory oil, and oregano. And we also tested Timeworks Act and Time Guard. And what we did was to use different concentration of each of these products amended to the culture media, and then we evaluated pathogen cultures or isolates, the growth of them on amended media. And for blueberries, we had 6 cholecalciferum or anthracnose isolates. This is one of the biggest problems in Florida and Georgia. We also had target spot isolates and stem blight isolates. And for peach, we had some brown rot isolates, 6 of them. The other 2 phases, before we put the formulated products in the field, we wanted to know how safe are they, because we were going to farmers' field, organic farmers' field. So we evaluated for crop safety for all of the products that we were going to try. Cinnamon oil cinerate was added in the 2nd year because it was a new product at the time. And then our 3rd phase was to actually set up trials on farm field assessment of these products. Really quick, the phytotoxicity trials, we evaluated for the low, high rates, label rates from each of the products. And we also evaluated the 5 times the highest rate. We were trying to gauge what's the range of safety for each of these products for the growers. So, we can give them a range that they can work within. Then, for the trials, they were pretty much the same. For blueberry, we actually evaluated 4 cultivars. We had 4 treatments for the first year. Cinerate was added as a 5th treatment, again, for the 2nd year. And then we had 6 applications pretty much every week, but from bloom to harvest. And for peach, we had the same. We only use 1 cultivar, US Sun. And we, again, The experimental design is the same, randomized block design. We had 4 treatments. Our control, we only use Time Guard for peach. And then we use our also or polyoxin D as the fungicide control. And then we had a mixture of Time Guard and also at the peach trial. For this one, the first year we had a kind of 2 weeks interval for each application. Then for the second year, we had a week interval, but just 6 weeks before, right before harvest. And then we went through fruit disease evaluation. These were similar between blueberry and peach. So we started with fruit collection, triage, especially for the blueberry, which we then surface sterilized in 10% bleach. And then we did about 7-day incubation. Then we score for disease incidence at day 3 and 6. So let's look at results really quick. So for our lab assays, this is a typical example of what you'll be seeing for the next few slides. Here we have the results for thyme oil for our anthracnose isolates, which we had 6 isolates. On the y-axis, you have the relative growth of each of the isolates. X-axis has the oil concentration. Thank you. Got it? Oh, thank you. Thank you. All right. And the x-axis has the oil concentration. And as you can see, as the concentration of oil increases, as the concentration of oil increases, the growth of the isolates decreases. It's kind of hard to see. The pointer stopped working. But the higher the concentration of oil, the less the isolates grow. So this is the same figure, but I wanted to show you that this trend is repeated for Thyme Guard, and Thyme Guard has about 23% thyme oil. And it's also repeated for oregano oil and savory oil. And these are for the same isolates. Now, we see the same trend for our target spot as well as stem blight isolates. These are also blueberry isolates. Again, as the concentration of oil increases, it's kind of hard. Let me see again. Yes. As the concentration of oil increases, the growth of the isolates decreases. And this is again repeated for thyme oil, Thyme Guard, oregano, and savory oil. For the same for the anthracnose isolates, when we look at Thymorex Act, on the other hand, we went all the way to the highest label rate for the product. And as you can see, even to the highest, you barely make it to halfway, 50% inhibition with this product. And this is across all 6 isolates. And then for the other blueberry isolates we look at, we see the same trend for most of them. And one of them actually seems to be, Thymorex seems to be helping it grow a little higher, better. So looking at, now looking at our peach isolates, we see the same trend again for thyme oil, higher concentration, low decrease in growth, Thyme Guard again, and oregano, and same thing for Thymorex, where it's really not inhibiting much. So overall, our results the, agree with previous studies, and they demonstrated that there is good efficacy in Petri plates for the products that are on the market. But also, Timeworks Act did not inhibit any of the isolate that we actually evaluated. So, for our phytotoxicity trials, what we observed was that for TimeGuard and Timeworks Act, for the 5x higher rates, we had burn-like symptoms, as you can see in these pictures, but no such thing was seen for the sin rate at 5x, 5 times a higher rate. And within the recommended label rate, everything else was fine. But that was until about the 5th application on the 2nd year during our field trial, which I have pictures that are coming up. Now, looking at our field assessment results, starting with the blueberry results. Overall, So overall, as you can see on this figure, we could not observe any treatment effects, and this is across all of the treatment. Not the treatment, but the diseases that we were looking at. So anthracnose, green mold, Alternaria fruit rot, nor rust, and this was for the first year. We observed the same trend for the 2nd year, except for anthracnose, and this was within the varieties. So for anthracnose, for the 2nd year, only for meadowlark do we see a treatment effect, but only for oso, which is not an essential oil product. And for peach, again, we saw no treatment effect for our first year, but we had some date effects, and this was repeated again for the 2nd year. So let's get to the take-home messages. So the commercial products were shown to be safe according to label instruction, but these are all products, so if you use them with caution. And as I mentioned before, these pictures are what we saw in the field last year, which was our 2nd year. This, for both of these instances, was at our 5th application. We used the same plants for both years, and this was a 2nd year application on the 5th application, so technically the 11th application. And although in the lab we have observed really good inhibition of the pathogens, the field evaluations for the efficacy of these products on both blueberry and peach demonstrate that the efficacy, at least of the products as they are formulated right now, and under the conditions that we have tested, not they pretty much don't work. So we do, however, see also as a potential organic fungicide alternative for anthracnose on blueberry. So for our ongoing investigations right now, we are looking at using these essential oil products more on the post-harvest disease management side. There's a lot more promising data on that side of things. So we have a student working on that. And then for more of the fungicide side, there is still need to be working on the formulation side of things if we're going to be using these products as intended as fungicide, because volatilization is a big part of essential oil antimicrobial efficacy. So we need to work on how can we reduce that while we are using them as we're spraying them in the field under especially hot and humid condition like we did. Okay, so thank you for your attention, and I just wanted to leave you with this. Again, please, if you use these, just be careful and make sure you understand that we do have great lab results, but in the field it's different. So, and if you have any questions, please go ahead. Thank you.
Thank you.
Yeah, I don't know if I missed it, but I know you said that you had one of the trials where you were spraying ozone in conjunction with essential oil. Is that
Yes.
on one of the graphs?
Yes, it was. Mm-hmm. Yeah, can you see it? It's, right. It's, we only did time guard and also for peach. So by the time, since we did the lab assays first, and TMRX didn't really, end up. So we only use time guard for peach. So, and what we were thinking of, like at the time, we have, again, we have collaborators from different units all around. They were seeing also also in pure spray actually, the combination as a possible good control for brown rot especially. And since we were going to be using that as our quote-unquote fungicide control, they wanted to see if we can try also and possible an essential oil, if maybe we'll have a synergetic type of, effect. So that's why we did the combination. Yes, exactly.
We have time for one more question. If anybody has any questions.
Yes.
Do you have a hypothesis as to why it worked great in the lab and not in the field, or are these things photo-degraded?
Yeah, so there is of that for the full, because it's been pretty hot, for sure. But we're thinking the volatilization aspect is really the biggest aspect of it, because like not only is it, we usually spray really early in the morning, we try it, but like these are oil products, and we can tell you like, especially for the blueberry trial, since we have 4 cultivars, by the time we end our 4th, by the end, if I go back to the first one, pretty much there's nothing there. It doesn't stay, so it's like totally gone already. So like, we need to work on that formulation if we're going to be using it as, sprays and essential. We need to have a way to make sure that the products stays longer and or like, is time release or something like that. So, yeah. Mm-hmm.
It sounds promising, but there definitely needs to be more work.
Yes, exactly.
Thank you.
Thank you.
All right, next we will have Dr. Jake Maurer come back up and discuss how cover crop mineralization influences gas emissions in organic production system. Greenhouse gas emissions in organic systems.
Okay, need to use the microscope again.
but I'll try to turn it back
Okay. Wow, the room really thinned out. Every time I turn around, Tim's a little closer. It's like you're sneaking up on me. Yeah. What, the volume's okay? Maybe a little louder. Okay. So, thank you for being here, all of you. I'm going to talk about cover crops, carbon, and greenhouse gases, and why you should care. I work with a lot of commodity crop producers. I spend a lot more of my time doing that than working with specialty crop, horticulture crop producers. But I think it's important to everybody, particularly because, well, we can put some more carbon in the soil. We can build its function up. But the federal government during this administration is offering sizable sums to farmers who engage in practices that are quote-unquote climate smart. And I think understanding the science behind this and developing it better is probably going to help them and help our farmers understand where their strategies are going to be best applied, right? So just a little primer, again, that the atmosphere is small here. The atmosphere is about 800 teragrams of carbon up there as mostly CO2. That the plants, all of the plant and to a lesser extent animal life on the planet that contains carbon is about 500 teragrams, but the soil can hold 2,400, right? So it's essentially 3 times as much as a storage organ for carbon on the earth as the atmosphere, and a good 5 times as much as all of the plant life living above the soil, right? So that's a pretty big difference. And so what we say is that if you can make a small difference in the soil by storing carbon there, then you are making 3 times the difference if you're taking it out of the atmosphere, right? Does that make sense? So there's been a lot of talk in my profession, soil scientists, for a few years about how do we get the agricultural practices outlined and in place where we can really make a difference, because it's soils that are being managed intensively that are going to make the difference. I mean, if you've got a forest, you don't want to change that. If you've got a corporate campus or school campus, you're not going to be changing that soil very much. You can change the way you landscape things for the better, but these soils that we're tilling, these massive acreages in this country and around the world, that we are missing opportunities to put cover crops or roots into in the off season. That's the low-hanging fruit, right? Does everybody get that? Make sense? Okay, so that's been our discussion for a couple of decades here. Like, how do we attack agriculture this way and put it all on the backs of farmers, right? But fortunately, there is some there is some money out there, and I would love for everybody to avail themselves of that, because we already know that we're not appreciated in agriculture for the things we provide: clothing, food, and we need to do a better job of sharing that with people. Don't hide our light under a bushel, etc. So for the next, we've got like 7 hours for this talk. We're going to talk about what are some of the things we grow cover crops for, what are greenhouse gases, and I'm going to cover an organic cotton study that we did for 3 years out in Texas, and what we learned about certain greenhouse gases under cover crops and not cover crops, and with poultry litter applied. So it goes to sleep, I think, every once in a while, the enter key. What are some of the things? We talked about this earlier. Was anyone in here when I was bloviating earlier? Okay, you were? All right, so the things, I really love them. The one thing they always do is stitch the soil together and prevent it from eroding. But we also often get with these roots better infiltration, better water storage capacities. If you love the bees, you love the butterflies, you can really provide a habitat for them, right? And a pathway for the monarchs, get the bees coming earlier. Most of our fruit and veggie production really relies on pollinators, right? You can also get involved in beneficial insect attraction. Predatory wasps and those kind of things can have their populations greatly increased by providing the right kind of habitat and attractors for them. We've got nitrogen, nutrient scavenging. I talked earlier about phosphorus, right? But just having living roots in the soil gives you biodiversity that we were talking about with the other attendee earlier. And so there's lots of different things, and I always urge people, as I said earlier, to know what do you want out of a cover crop. And this is really going to affect what you pay for, what you choose to plant, when you plant it, and what your strategy is with cover cropping. Because a cover crop's not a cover crop, it's not a cover crop. There are different strengths in this one, different strengths in this one, different strengths in that one, right? So when we're talking about the carbon cycle here, the earth, the atmosphere is one big lung, right? There's CO2 going up. Plants are taking it in. They're fixing it into the carbon that provides all the structure for their cells. And so again, here we've got about 600 in all plants, 800 petograms, not teragrams, excuse me, in the atmosphere, and 2,400 in the soil. These are generally accepted and frequently published amounts, and we all sort of agree on them. So to give you an idea of what a petagram is, it's 10 to the 15 or grams, or I'm not going to read all those zeros out, but it's a lot, right? It's more than you can think about at once, but it's about 1.1 billion tons is a petagram, right? So if you've got 600, 800, 24, you can now sort of picture that in your head without it exploding, right? Okay, so that's a lot of carbon that can be shifted from one place to another. And there's been a lot of interest driving carbon soil sequestration, soil carbon sequestration to improve degraded soils, reduce erosion, improve loss of fertility, restore organic matter. We have this idea, and I think it's right to say this, that a soil with very little organic matter in it performs less well than a soil that has a decent amount. What's the right amount? People differ in their opinions on that, but we know a degraded soil that doesn't produce very well is one that doesn't have very much carbon in it at all. Climate change predictions are out there, whatever you feel about it. It was a hot summer at my house. Like one of the farmers I was working with said when he got off the combine, it felt like somebody just turned a hairdryer into his face. And then I couldn't get that out of my head for the rest of the summer, because every time the wind blew, it was like someone had just turned a hairdryer on me. So small changes in soil carbon are going to result in larger changes in atmospheric carbon. Agriculture is a fairly large emitter of greenhouse gases, so it's also low-hanging fruit for that. Agriculture uses a lot of petroleum carbon to get the job done, right? And there are ways that we can talk about reducing that, but not in this talk. So carbon or climate market payouts, it's going to depend on the Farm Bill. Hey, we strung the budget out a little bit longer a couple of weeks ago, earlier this week, right? So federal funding does exist for climate smart practices, and it's out there now. And so it can be applied for. And we're getting that in research and extension to provide better answers to farmers who have questions. So NRCS can be a partner for that. They're FSA, Farm Service Agency, and the Natural Resources Conservation Service can be partners and can be approached and worked with to offset the cost of trying to solve the climate problem as a farmer. So, I heard about this, 15, 20 years ago. Biochar was being talked about, and somebody I used to work with over here at UGA, Julia Gaskin, had a project in that. And so that kind of stuck in my mind, and I'd read a little bit about it. And does anybody know about the terra preta soils in Brazil? Have you heard about this? Well, this Dutch soil scientist went down there. He's an agriculturalist, and he noticed that mango trees over here were growing just like big, right? That wasn't a big vocabulary word, and over here they weren't growing so well, right? But they seemed to be in these circle patches where the mangoes were just thriving. And he had this brilliant idea. He was like, why don't I just dig a hole and look at this? There's a difference in the soil. And they found that there was all this black soil beneath the thriving mango trees, and the other stuff looked like this, right? It looks like good Georgia dirt, right? Anyway, so loss of seed is associated with yield declines and increased fertilizer. We got this idea through this guy's work that maybe we could add carbon to soil and see these kind of changes, or we could add biochar, which is a particularly long-lasting type of carbon. But also keeping the soil where it belongs, right? So erosion is removing the top inch of the soil, the top half inch. Each year you don't protect it. That's where most of the carbon is on our farming soils. And so we're throwing that away. You own that dirt. Keep it where it belongs, right? And then there are other soil properties that are reliant on soil carbon, including aggregate stability, water capture and retention, which are physical properties, but also bacterial and fungal communities eat carbon, okay? Well, most of them do, heterotrophs do. They eat carbon, and if it's there, you will have more and more diverse organism communities. But you can see here how the corn's doing there and how it's doing here over this. So that idea has maybe run ahead of itself, run amok. I mean, there's lots of biochar being sold for potted plants today, and people are scaling up for agriculture, but it doesn't take as much as most people think, but it's very light and can blow away very easily. So biochar has been interesting to work with. Here, we're not going to be putting biochar into the soil with cover crops, right? So we're talking about the different greenhouse gases that are different segments of our society that are responsible for greenhouse gases. Agriculture is about 11% of the overall greenhouse gas, but it's the one probably we're going to have the most success, at least in the short term, in moving the needle, because no one wants to give up cars and have mass transportation. Industry is responsible for all of the things that we have around our homes. So we're focused on agriculture, right? And then the thing that I need to mention is we've been so focused on carbon dioxide and putting carbon in the soil for a long time that we've forgotten there are two other really important greenhouse gases to keep track of. And one of them is methane, which the current estimate for its carbon dioxide equivalent, in other words, for every molecule of methane you have, it's as effective at trapping heat in the Earth's atmosphere as 28 molecules of carbon dioxide, okay? Because it breaks down, it has a half-life of 12 years. It's not its lifetime. But over 100 years, they say that it's 28 times. But immediately, like as soon as that molecule's released, it is 87 times as potent as carbon dioxide. And so there's a little bit of, I like to use that in my messaging, right? Right now, every methane molecule that is released is 87 times as potent as carbon dioxide. And what's worse is this nitrous oxide guy here, which is almost 300 times as potent at trapping heat within the atmosphere as carbon dioxide. So we often get lost in our discussions about carbon dioxide and putting carbon in the soil, but are we paying attention to the other greenhouse gas emissions that might be associated with the recommended practices that we're recommending, right? And so if we look at their emissions over time from 1991 to 2021, we can see, I mean, they're fairly stable. They're increasing. They're not going down. But we are seeing a lot of nitrous oxide going up as well, and it's 300 times as potent as CO2. So these are things that I've been keeping in mind when we make our measurements. Hey, if we're putting carbon into the soil and we're reducing CO2 emissions, are we really reducing the total equivalent of greenhouse gases by doing this, right? So a team of us have been doing this research for 3 or 4 years, and we're finding some really interesting counterintuitive things about that. So soil is a massive habitat for microorganisms. They're doing all this work to break down the carbon from plant tissue and turn it into CO2, going back up into the atmosphere again. There's a lot there. You know, we were talking in the earlier talk that there are 1 to 2 tons of bacteria per acre in an acre furrow slice. It's like 10 billion cells per gram of soil. That's a lot. That's a low ball. Some people say 100 billion. Just like crop rotations, diversity is key. So the number of organisms isn't as important as the number of functional niches they fill. And carbon is the food source for most of these microorganisms. So we're getting their benefits when we have carbon in the soil, and a lot of that comes from root biomass, plant top incorporation, root exudates. I always talk to my audiences about the type of carbon too, because it's not just that we can put carbon in the soil, how long is it going to stay there, right? The longevity of that carbon makes a difference when we're talking about carbon sequestration. So if I threw a bag of sugar in the soil, it's probably going to last 2 hours to 2 days. Those microbes are going to chew it up really, really quickly, right? It's very easy carbon to break down, but things like lignin, biochar, cellulose, and hemicellulose have longer lasting humus, longer lasting lifetimes in the soil. And so we need to think about things that way as well. And it's always going to depend on environmental conditions. So where I work in Texas is hot all year round. We have warm enough, most of the southern part of Georgia, the winters are warm enough that bacterial activity is working on breaking down the stuff all year round. In Minnesota, on the other hand, they get a reprieve all winter, right? So we've done some work on just looking at how much biomass different species of cover crops can produce in my area. And so, you know, what are you growing cover crops for? If you want to put carbon in the soil, you want to capture CO2, put it into plant mass, put it into the soil, then biomass is your thing, and you want big biomass. And so we found certain things like, well, let me just read it here. The highest potential are things like sorghum Sudan grass, sun hemp for the summer, sunflower is actually a lot of biomass. And in the winter, we're finding things like cereal rye, mustard produces a lot of biomass for us, and it will across the south, and oats and winter wheat and triticale are pretty good at having a large biomass potential. So if you're looking for putting carbon in the soil, either through roots or through green manuring, those are the ones you might want to focus on. Okay, so I'm going to move on to the specific research project that we did for 3 years on organic cotton, and was working with the same weed scientist I mentioned earlier, and his idea was to look at defoliants and weed control that would be organically approved, because where we're at, you have to defoliate in order to harvest the cotton, right? You know what that is. But we were monitoring the carbon flux as affected by management practices, by tillage type and cover crop type. So we had 2 tillages, full on, all the tillages, tandem disc followed by harrow, and then we had no-till as well. And we had cover crops, the 3 species in a mix. We had a weedy check where we did nothing to control the weeds, and we had a weed-free check as well. So sort of like a negative and a positive control. And then we were looking at CO2 evolution, but also measuring nitrous oxide and methane as well. Had a split plot design, and does anybody care about the experimental design? Okay. Look, we had 2 types of tillage, 3 species in a mix, and then a positive and a negative control with a weedy check and a weed-free check. We did a lot of measurements for CO2, nitrous oxide, and methane, the 3 most important greenhouse gases in agriculture. And then we found, in 2021, which I think is the data, that's the data I have here, we can see that under minimal till, we had lower amounts of biomass than with conventional till. This isn't something that the NRCS says a lot to farmers, but in my area, if we till, we get plants to grow bigger. It's just something about these types of clays that we're growing that they do need to be broken up if yield is your goal and biomass is. But in a conventional till, we had almost 5,000 kilograms per hectare, and then the mix, which also included oat, had 3,600 kilograms per hectare. Again, this is the highest up here in minimal till was pea, and the weedy check had quite a few weeds in it, right? So that's what we were looking at. We added poultry litter, did it on a carbon basis here. But all of these are on a carbon basis. So the total carbon added in our experiment is here. We backed off the amount of poultry litter for the legume monoculture, which is what we would recommend for farmers. And then, yeah, some differences there. We put them, had a grad student working in the lab, doing some incubations. We got cracker jars from the Websterant supply place and fitted the tops with septa so that we could take syringes out of the head space and put it on a GC and get the greenhouse gases. We also did some traps where we trapped CO2 and then measured it directly so that we could have more idea about that one. Traps don't work for N2O and methane. But we did this for 90 days, and then we measured the CO2, and we found that about half of everything that we put down as carbon was blown off over that 93-month period, okay? With the weed-free check, we had 64%, but there wasn't as much biomass there. But what was really interesting to me is here's another example of we have a recommended practice, a best recommended practice for soil health, for soil building, carbon of no-till. But we had more carbon blown off under no-till than we did, or minimal till, than we did under conventional tillage. All right, so by incorporating or green manuring our crop residues, we had significantly and practically lower rates of CO2 consumed by bacteria and blown off into the atmosphere. And you can argue that we probably didn't have as many bacteria because we destroyed their habitat by tilling, but the fact is, if we want to put carbon in the soil, that is counterintuitive to what we've been talking about for a long time in terms of conserving residue from cover crops, right? It's an interesting thing. We fit the data for all these time points to a first order model so that we could generate some estimates of the total amount of mineralizable carbon that we could get, and that's where we came up with these mineralizable potentials here. And you can see these are on the same scale on the y-axis, which is cumulative CO2C evolved in the traps, and then days of incubation on the x-axis. And you can see that here in the conventional till that these are lower curves. I mean, we're evolving less, just like the numbers in the previous slide said. So that was counterintuitive. So we want to be able to report that and maybe figure out how we can control an unwanted outcome from a best recommended practice in the future. Moving on to the methane measurements, which we took head space out with syringes for this and put it on a GC analyzer. And what we know about methane is that soil's generally a sink for methane. In other words, soil will absorb methane. So that's actually a pretty good thing, right? If you've got a greenhouse gas and it's somewhere at the soil-atmosphere interface, the soil might grab it and turn it into carbon. There are methanotrophs which eat methane and take it away, basically. When soils are aerobic, this is the case. When they get waterlogged or they go anaerobic for some other reason, methane generally is produced rather than absorbed. And that's why flooded rice culture around the world is responsible for 16% of the greenhouse gas emissions. So that's it's pretty interesting. Anyway, what we've got here are, I want to walk away from the microphone, time series, individual sampling events, and we can't do cumulative here because we're not really trapping it all. We're just taking like little snapshots at a time. And you see the sort of trends we have, maybe a jump here at 25 days. We see that in 2021 we had a decrease or more absorption as we went further and further along. And this was true for some of the treatments here, but at the bottom we've got a bar chart which is inverted to show us how much we're absorbing. And what we see is that when we green manure or incorporate into the soil, for the just poultry litter alone, we see more methane absorbed when it's placed on top, right? But for poultry litter plus oats, we see more evolved or more absorbed when it's unincorporated. So there's some back and forth results that we're seeing here. There are occasional significant differences, particularly in 2021 when we're looking at the poultry litter versus Austrian winter pea, where we're seeing a lot more absorbed when incorporated than laid on the top. So this is telling us that maybe we should be looking at these differences to see how much methane we can maybe store into the soil, or how much of this effect can we harness, right? So, and then nitrous oxide, well, we see in 2020 pretty flat-lined, and then as we get into a little bit later in the year, we see that we've got a jump around 20 days. It's not a huge jump, but again, this is 273 times as potent as CO2, so any release needs to be taken seriously. In 2021, we see a jump right away, which is probably in the first week or so related to the most nitrogen being mineralized or broken down from its organic forms into its inorganic forms, and that process is known to release a little nitrous oxide. Down at the bottom here, the bars are going up because it's a release rather than an absorption, right? So we see differences again in incorporation into the soil versus surface applied. We're seeing generally in 2020 more, most of the time when it's surface applied, nitrous oxide is being released than if it's incorporated. And then in 2021, on the right-hand side on the bottom, we are seeing mostly the same thing, except for with the poultry litter that's incorporated, it is more responsible for nitrous oxide emissions. So there are some differences there that we're continuing to look at in different cropping systems, year in and year out, cool seasons, warm seasons. But it does appear that the type of cover crop is important for all of the the three major greenhouse gas emissions. Legumes, now this is the thing, like we want them to put down nitrogen, right? If we have fixation of nitrogen from legumes and deposition into the soil, well then we can really back off, or maybe not even have to buy any synthetic fertilizer, or in organic situations apply so much organic fertilizer. So that can be very, very good. But what happens when legumes break down, because they are much more rapidly broken down than things like oats and mustards, they could get ahead of themselves, and they can start to get oxygen limited by that breakdown process, and we get into anaerobic situations at microsites in the soil where, unfortunately, nitrous oxide is the product rather than ammonium or nitrate. So that's what we're seeing. We're drilling down on this. A couple of us in a team that are working on this are really looking at different aspects and trying to understand better what's happening. But I'm convinced that during nitrification, which is the conversion of ammonium to nitrate, that we are getting ahead of ourselves when it's too rapid. We're getting anaerobic at microsites, losing that oxygen, and then producing nitrous oxide instead of nitrate. And we don't want that. So I'm beginning to believe that a monoculture of legumes is probably not the greatest thing for one of the greenhouse gases, right? So a mix can slow that down, keep us from getting ahead of ourselves, and release a lot less nitrous oxide. So cover crops increase soil carbon, yes, but which species is going to have the most biomass? I prefer root biomass over top biomass because it has more lignin, more suberins, and tends to be slower to break down. So if we're trying to put in carbon, maybe not the above ground biomass is the thing we should always focus on, but also something that has good below ground biomass. I have seen this in the literature, but more often, research in the past has shown that no-till releases less carbon dioxide than till, right? But in our experiments, we were clearly seeing in almost every comparison that no-till was promoting more breakdown of carbon from crop residues and poultry litter than when we tilled and incorporated, right? And so then the last thing here is that nitrous oxide can be released in greater amounts when nitrogen is supplied in easy to mineralize forms, okay? And so we've been playing around with this. We've got that GC analyzer, and we've got boxes and boxes of syringes, and we're just playing around with this and seeing what's going to pop out and what kind of recommendations we can do. We've had some input into the farm bill. Whenever that gets passed, I don't know. You have any insight on that? Okay. We were asked to provide some input on what the next best recommended practices list should be for climate smart agriculture, and we've been kind of repeating this thing that mixes are probably going to be better for reducing N2O emissions than legumes monocultures, right? So I may have talked a little faster than my Georgia accent was supposed to go, so it looks like this is my last slide. We have plenty of time for questions, right? Okay. Ask some questions, please, because I probably forgot to cover something. It's all up here. Yes, in the back, sir.
Sorry, I missed the beginning of the presentation.
Okay.
Were you only looking at cotton, or were there some other cotton?
This was a 3-year cotton study, but we have we have research on wheat, what else? Sorghum, corn, and yeah. So the big commodities that we grow where I work, and yeah, and forages too. So pasture production, hay fields. I've also looked at cool season overseeding in that as well. So.
Gotcha. A follow-up question. What else would be in the rotation, or what else might have been in the rotation for the cotton field?
So this, we did what we're not supposed to do, cotton, 3 years in a row, because it was about trying to understand defoliation and weed control better. And I was on to round out some of the things that we were looking at from the soil standpoint. What are the other benefits of cover crops? And so that's what I focused on. But normally a rotation would be something like corn, cotton, or we would have sorghum in there. And occasionally, depending on where you are in the state, you might go with wheat over the cool season and then skip a year and put in cotton the next year. So those are typical commodity rotations for us, yeah.